Okay, so let's go over the first exam. It's going to be 50 questions long. It's going to be mostly multiple choice. I may have one or two true false in there. Now, this first exam is pretty heavy on material. We're going to cover 6 chapters, okay? 1 and then 4 through 8. And this is a closed book exam. Now, for those of you in my online and my hybrid classes, you will be taking this exam on your own device, and you'll be using the proctoring software called Proctorio. And Proctorio requires a webcam. So I want to make you aware of that well in advance so you can secure a device that has a webcam. If you are in my face-to-face class, you will come to campus on the day of the exam and take it in person. Please remember, this is a closed book exam, so no notes will be allowed, and any looking at notes or asking anyone next to you or looking at anyone next to you's tests, anything like that, any kind of irregular behavior is going to be considered cheating, and you will make a 0 on the exam. Now, I do drop the lowest exam grade as long as you take the final exam in this course. And so if you don't do as well on this first exam, that's okay. The first exam is often the hardest. You're getting used to the content that I'm teaching over as well as how I teach. So there's generally a learning curve on the first exam. And for many of you, exam 1 is going to be the low exam grade that gets dropped. And again, that's okay. I'm just saying this to try to reassure you not to panic if you don't make the grade that you want to make on this first test. Okay, so let's dig into the content of the study guide. The first question here is, what are disease-causing microorganisms called? This is a simple definition. Just remember that if the microorganism causes a disease, it's called a pathogen. Remember also that most microorganisms, the vast majority, are not disease-causing. Only a relatively few are pathogens. Okay, microorganisms that lack a nucleus, these are going to be called prokaryotes, okay, which means pre-nucleus. And the prokaryotes are going to be bacteria and archaea, 2 out of 3 of the domains of life. Remember that what makes eukaryotes different from prokaryotes is that eukaryotes have a nucleus, right? Eukaryote means true nucleus. They also have membrane-bound organelles. So that's going to be organelles like the mitochondria, lysosomes, the Golgi apparatus, the endoplasmic reticulum, both rough and smooth. Okay, so all of those are membrane-bound. You need to be familiar with the steps of the scientific method, which is, in science, how we try to answer questions that we don't know. So the first thing that you do is you observe something that interests you. Next, you ask a question about what you observed. One such question may be, why does this phenomenon happen? Next, you're going to form a hypothesis, and all a hypothesis is it's a tentative explanation for why something happens the way it does. Now, your hypothesis has to be testable. So your next step after forming your hypothesis is going to be designing an experiment to test the hypothesis. Then you're going to gather data and analyze that data. And you're going to come to a conclusion on whether or not the data from your experiment supports your hypothesis or disproves your hypothesis. Your hypothesis not only, it needs to be testable. Not only does it need to be provable, if true, but it needs to be able to be refuted as well in order to be a good functioning hypothesis. All right, next. Microorganisms are best described as, and the definition we use is whether or not you need magnification. So microorganisms are going to be described as organisms that are too small to be seen with the unaided eye. In other words, you have to use magnification. All right, who was the Dutch linen merchant that made and used quality magnifying lenses to see and record microorganisms? This is just a little bit about the history of microbiology. We have a lot of interesting figures in early microbiology, and this guy is Antony van Leeuwenhoek. He developed microscopes in order to view the threads on his linens, and one of the things he did in his spare time was he looked at things like teeth scrapings under his microscope and discovered that there are tiny living creatures in just about everything. What is spontaneous generation? Well, remember, this comes before the era of microscopes, and this is when people believed that living things arose from non-living things. Of course, then with the advent of the microscope, we learn that there are tiny microorganisms, living organisms that are invisible to the naked eye, and it's from those organisms that other organisms like them are derived. So you get things like cell theory and the germ theory of disease. All of that comes after the invention of the microscope and disproves the belief of spontaneous generation. Okay, what are Koch's postulates used for? And you may recall Koch is another gentleman from early on in microbiology. And what he did is he came up with a series of logical steps, that's these right here, that essentially tell you whether or not a microorganism causes a certain disease. And I'll just walk you through kind of what Koch's postulates are. So first, you want to, you're looking for a suspected pathogen that's found in abundance in diseased animals, but is not found in healthy animals. So that's your first step. Secondly, you got to get this microorganism, the suspected pathogen, and you need to isolate it and culture it or grow it, okay? Next, you take this organism that you've grown, that you've cultured, and you introduce it into a healthy animal or healthy organism and witness its effects on the organism. And then you have to get that pathogen, that microorganism, out of the diseased animal again, and essentially identify it and make sure that it's identical to your suspected pathogen. If you perform this series of steps like so, you can prove that a microorganism is the causative agent of a particular disease. All right, next, know the order of taxa from most general to most specific. So remember, taxa are going to be the domain, the kingdom, the phylum, the class, order, family, genus, and species. To remember this, I use the mnemonic, Dear King Philip came over for good spaghetti. Now you're going to be asked to order these from most inclusive to least inclusive. So what does that mean? Well, the most inclusive is going to be the taxon that includes the most organisms. So that's going to be of these given taxa, right, of all the given taxa, that's going to be domain. Domain includes the most organisms. The least inclusive taxon is going to be species. It includes the fewest amount of organisms. I want you to be able to recognize a scientific name. You're going to be given a variety of names, and you need to be able to tell me that the name looks like this. So it's going to have the genus capitalized. It's going to have the species all lowercase, and then the whole thing is going to be italicized. All right, that's what a scientific name looks like. Okay, what gives spirochetes their twisting locomotion? Now remember, spirochetes are kind of these coil-shaped bacteria. They're always gram-negative, and they penetrate tissues by using sort of a flexing, twisting motion. Now the way they're able to do this twisting motion is they have flagella on the inside of them. So these flagella are called periplasmic flagella, and they're found in the periplasmic space. And the periplasmic space in a gram-negative organism, such as a spirochete, is going to be between the cell wall and the outer membrane. That's where these flagella are located. And when they contract or move, they make the organism, the spirochete, kind of wriggle. And this wriggling motion, this twisting motion, is what makes spirochetes so good at penetrating through tissue. All right, I expect you to know the differences between the bacterial appendages. So this is going to be cilia, flagella, fimbriae, and sex pili. All right, remember that cilia are going to be found in eukaryotic organisms, some eukaryotic organisms. And they're essentially small projections of the plasma membrane that beat in unison and help move substances across them. Flagella, on the other hand, this can be found in prokaryotes and some eukaryotes as well. This is an appendage for locomotion. All right, so remember we've got the bacterial flagellum, which rotates and causes the bacterium to move in a series of tumbles and runs. Eukaryotic flagella are built a little bit differently, move a little bit differently, but the overall point of it in prokaryotes and eukaryotes alike is that the flagellum allows for the organism to move. Okay, remember that fimbriae are short, bristle-like appendages emanating from bacteria, and it helps the bacteria adhere to, for instance, the surface. And then a sex pilus is going to be a rigid tubular appendage through which DNA is transferred during bacterial conjugation. Now, we learned that bacteria, they reproduce asexually through binary fission. So you would think there's not much genetic variation among bacteria. However, the sex pilus allows bacteria to transfer genetic information between one another. It's not a form of reproduction, but it does introduce new genes into different bacterial species. So sex pili allow for genetic diversity among bacteria. Okay, you need to know the flagellar arrangements. So one flagellum coming out of one pole of the organism is going to be monotrichous. If there's a tuft of several flagella coming out at one end of the organism, that's going to be lophotrichous. If there's flagella coming out of both poles of the organism, that's going to be amphitrichous. And then there can be flagella emanating from all parts of the surface, all over the cell. And when that occurs, it's called peritrichous. All right, the outcome of the Gram stain is based on what? Well, remember, the Gram stain stains differentially between gram-positive and gram-negative organisms. The gram-positive organisms are going to look purple. The gram-negative organisms are going to look red. And the reason for this is because they have differences in their cell wall structure. Remember that gram-positive organisms have a very thick peptidoglycan layer of their cell wall. So they're going to hold on to that primary stain in the Gram stain. They're going to hold on to that crystal violet. Whereas the gram-negatives have a very thin peptidoglycan layer. And so they're going to lose the crystal violet and take on the safranin. when you counterstain them. So that's why the gram-positives look purple and why the gram-negatives look red. Okay, what are bacterial endospores and what are they used for? Well, remember, a bacterial endospore is a highly resistant, dormant form of the cell. So it contains all of the genetic instructions of the bacterium. And what happens is these endospores are formed under harsh conditions. So when the environment becomes unfavorable, the bacteria produces endospores, which can remain dormant indefinitely for millions of years. When environments become favorable again, when nutrients and water appear again, the endospores germinate and become vegetative cells again, become regular bacteria again, okay, and go on living their lives. I want you to know that endospore producing bacteria have a two-phase life cycle. You have the vegetative cell. This is the metabolically active bacterium. And then you have the endospore. This is that inactive, dormant, incredibly resistant form that can survive just about anything. Very low acidity, very high temperature, dehydration, UV radiation, all of that. These endospores can survive. Now you need to know that the formation of endospores, that process is called sporulation. Remember that sporulation, these are not spores like they are in fungi. Fungi use their spores generally for reproduction. In bacteria, endospores are not reproductive. They are for survival. So you've got sporulation. This is going to be the formation of endospores. And then when the environment becomes favorable again, you're going to get germination. And that's when the endospores return to their vegetative growth. So they, transform from their inert structure to the growing, metabolizing, normal structure of the bacterium. Okay, what kind of microorganisms can live in extreme conditions? So you may recall that we've mentioned thermophiles, we've mentioned halophiles. So thermophiles love heat, halophiles love high salt content. Any kind of environment on Earth, you can just about expect has a microorganism that loves that environment. Even environments we think to be uninhabitable. Now, the microorganisms that are most well known to be extremophiles, they like extremes in the environment, are going to be the archaea. So on the test, you're going to see a question that gives you some qualities or characteristics of an organism. And those characteristics are that the organism essentially can survive in a very inhospitable environment. And it's going to ask you, what do you think this organism is? And the answer is going to be, it's probably an archaean. All right, what is the main component of the cell wall of fungi? We'll go back and recall that fungi are eukaryotes, but they do have a cell wall. But unlike the prokaryotes, the bacteria, their cell wall is not going to be made of peptidoglycan. It's going to be made of chitin. And the cell wall of bacteria, again, is peptidoglycan. The cell wall of archaea varies, but it's neither peptidoglycan nor chitin. Okay, eukaryotic organelles, you need to know the functions of them. So lysosomes, remember, this is a membrane-bound organelle, and it is responsible for intracellular digestion of food particles and debris. Mitochondria, again, another membrane-bound organelle, and its inner membrane contains enzymes that are involved in oxidative phosphorylation. Remember the folds of that inner membrane is they're called cristae, and it's on the cristae that the enzymes of the electron transport chain are, and this is where energy is made. Next, you have the Golgi apparatus, and essentially what the Golgi apparatus does is it takes proteins that the rough endoplasmic reticulum have produced, and it sorts them, it modifies them, and then it ships them out to other parts of the cell or to outside of the cell. Remember that the Golgi apparatus is membrane bound as well. And the nucleus, of course, is another membrane bound organelle. And this is where two things are located. So in the nucleus, you have obviously the genetic material of the organism, the DNA, which is the set of instructions for the cell to work. You also have the nucleolus. And the nucleolus is where ribosomal RNA is made and where ribosomes are assembled. Now going on to ribosomes, what do ribosomes do? Well, this is not a membrane-bound organelle. Remember that prokaryotes have ribosomes, and ribosomes are the site of protein synthesis. They essentially act as a table on which the mRNA transcript gets read and converted into an amino acid sequence. So they're the table of translation. The rough endoplasmic reticulum is the site of many ribosomes. So this is where a lot of proteins get made in the cell. And then you've got the smooth endoplasmic reticulum, which lacks ribosomes. That's what makes it smooth. The smooth ER is involved in lipid synthesis and especially in some cells in detoxification. Okay, on which organisms are cilia found? Well, cilia are only going to be found on relatively few organisms. Some protozoa, not all of them, but some of them, and then some animal cells. For example, human respiratory epithelium is ciliated. And the reason for that is the cilia in your respiratory tract beat in a uniform fashion, and they move mucus, which has trapped dust and pathogens, up your respiratory tract so that you can cough it out and remove that dust and remove those pathogens from your lungs. All right, know the difference in the prokaryotic ribosome versus the eukaryotic ribosome, and I've put the difference here. So remember, ribosomes are going to be composed of two subunits. There's the small subunit and the large subunit. These subunits come together to form the ribosome. Now in prokaryotes, the small subunit is 30S, the big subunit is 50S, and when they come together, they create a 70S ribosome. This is microbiology math. There is a reason why these numbers don't add up, but you don't need to worry about that reason. Just know that prokaryotes, so for instance bacteria, are going to have a 70S ribosome. Eukaryotes, on the other hand, have a 40S small subunit, a 60S large subunit, and when they come together, it creates an 80S ribosome. The way I remember this is eukaryote begins with an E, even begins with an E. These are even numbers, right? So that's how I remember that the 80S ribosome belongs to the eukaryotes. Now, why does this matter? Well, you'll learn a little bit later in this course that one of the common targets of antimicrobial therapy or antibiotic drugs is going to be the ribosome. So if you can break down the ribosome of the bacterium, you essentially halt protein synthesis in the cell, and that stops the cell from being able to grow and reproduce. However, you don't want your medication to target your own ribosomes. So the antibiotic drugs that we use target specifically 70S ribosomes, sparing our ADS ribosomes. This means our cells get to continue functioning normally. All right, next I want you to know the characteristics of fungi. So remember that fungi have cell walls. They're going to include single-celled forms, right, the yeasts, and filamentous forms, or the molds. Fungi are heterotrophic, so they feed on other organisms. Specifically, they tend to be saprobes. They feed on dead and decaying organic matter. They utilize a wide variety of nutrients. And importantly, fungi are not photosynthetic. All right, the organism in which adult helminths mate is called what? Well, remember, helminths are going to be our parasitic worms. And they have an interesting life cycle that can involve an intermediate host, which houses a larval or a cyst form of the helminth, and a definitive host, which houses the adult form of the helminth, and those adult worms sexually reproduce inside of the definitive host. So the answer here is definitive host. All right, you need to know which organisms have a cell wall and which ones don't. So bacteria and archaea have cell walls. Fungi have cell walls. Algae have cell walls. Protozoa and animals do not. You need to know the membrane-bound organelles and which organisms they are found in. So remember, we talked about this already. Membrane-bound organelles are going to include things like the nucleus, the endoplasmic reticulum, both rough and smooth, the Golgi apparatus, lysosomes, and mitochondria. And membrane-bound organelles are only going to be found in eukaryotes. Okay, next, what are cytopathic effects? Well, recall that cytopathic effects are going to be the virus-induced damage that happens to the host cell that you can see under a microscope. Things that you can see include changes in the shape, the size, and the orientation of the cells, if the cells had orientation. You can see development of intracellular changes, such as inclusion bodies. And these are basically little compacted masses of viruses and damaged organelles. You can also see syncytia form, and syncytia are going to be giant cells. And what happens here is multiple damaged host cells come together and fuse into a large cell containing multiple nuclei. And we call those giant cells. So cytopathic effects, this is just the virus-induced damage to the cell that you can see with a microscope. Okay, are viruses referred to as living and dead? Why or why not? Well, recall, we don't refer to a virus as being alive or being dead. We refer to it as being active, that is able to infect, or inactive, that is unable to infect the host cell. Why do we not consider viruses living? Because they cannot independently reproduce. They can't make more copies of themselves without using a host cell's machinery. Which organisms can viruses infect? All of them, okay? Can infect bacteria, they can infect protozoa, they can infect algae, fungi, and of course animals. They can infect plants as well. What does the genetic core of every virus contain? Remember, it's going to contain nucleic acid. And that nucleic acid is either going to be DNA or RNA, but it's never going to be both. You need to know the difference between naked and enveloped viruses. Remember, naked viruses only consist of that nucleic acid core and a capsid. That's it. The enveloped viruses have that nucleic acid core and the capsid, but they also have this envelope surrounding them which they derive from their host cell's cell membrane. I want you to know the viral multiplication cycle for bacteriophage specifically, and I've given it to you here. You're going to be asked to order the steps of viral multiplication. So first, you're going to have adsorption. Remember, this is where the virus binds to receptor molecules on the host cell. Next, you're going to have penetration. This is when the viral genome enters the host cell. Now, remember, this is a bacteriophage, so you're not going to have the uncoding step. Next, you have replication, where the viral components are produced. You have assembly, where the viral components are assembled. Then the virus matures, and then finally, it is released from the cell. All right, know the characteristics of viruses. The characteristics I want you to remember are that they are ultra microscopic, right? So you have to use something with higher magnification than a simple light microscope, such as an electron microscope. They have the ability to infect host cells. They take on unique shapes. They have genes, right? Genetic material containing genes that have the instructions for making more viral components, and they lack a metabolism. You should know osmosis. Remember, this is going to be the diffusion of water across a semi-permeable membrane. I have an example here. You have the semi-permeable membrane. You have a highly concentrated solution on the right-hand side and a less concentrated solution on the left-hand side. Another way to look at it is there's a higher water concentration over here. And water is going to move from the area that's least concentrated solution to the more concentrated solution. Or the other way to think of it is water is going to move from the high water concentration side to the low water concentration side until an equal concentration is achieved across the semi-permeable membrane. What is a heterotroph? Well, remember, a heterotroph is going to be any organism that has to derive its carbon from other organisms. All right, what is chemoheterotrophy? Well, this is a little bit more specific term than simply heterotroph. Heterotroph is going to be an organism that gets its carbon from other organisms. A chemoheterotroph gets its carbon from other organisms, and it gets its energy from those carbon-containing chemicals. For example, glucose. So an example would be aerobic respiration. Organisms that undergo aerobic respiration get their carbon-containing chemicals, like glucose, from other organisms, and that's an example of chemoheterotrophy. What are saprobes? I think I might have mentioned it earlier, but saprobes are going to be organisms that derive their nutrients from dead organisms. An example of a saprobe is a fungus. You need to be familiar with the hypotonic, isotonic, and hypertonic conditions of cells that have cell walls. So remember, in an isotonic solution, if you take this cell and place it in an isotonic solution, there's going to be no net movement of water in or out of the cell. If you take that same cell and you place it in a hypotonic solution, because of osmosis, water is going to want to diffuse into the cell. Now, in organisms that lack a cell wall, this can result in the organism bursting, rupturing. However, in bacteria, the cell wall helps the bacteria maintain its shape. In a hypertonic solution, water is going to diffuse out of the cell into the surrounding fluid. And the cell membrane is going to shrink away from the cell wall in a process known as plasmolysis. So in blank solutions, The cell wall helps prevent the organism from bursting. We said that's going to be hypotonic solutions. In blank solutions, there's no net change to the organism. That's going to be isotonic. And then in blank solutions, the organism's cell membrane shrinks away from the cell wall. That's going to be hypertonic. You should be familiar with all three of these conditions. Okay, what type of energy is converted to chemical energy during photosynthesis? Well, photo means light, so it's light energy that's going to be converted to chemical energy. What kind of environments can facultative organisms survive in? Remember that facultative means it has the option. So, for example, a facultative anaerobe means that it can use oxygen or it can choose not to use oxygen. And what that means is that facultative organisms can, generally speaking, survive in a wide range of conditions. Okay, growth factors. What are growth factors? You need to know. They are going to be compounds that the organism cannot synthesize itself because it lacks the mechanisms to synthesize them. So it has to obtain these growth factors from its environment. Okay, in a chemical reaction, how are reactants converted to products? This sounds really daunting, but it's a really simple answer. During a chemical reaction, reactants are going to be converted to products by either breaking bonds or forming bonds, or a combination of breaking and forming bonds. I want you to know the characteristics of enzymes. All right, so they're going to be catalysts. That means they increase the rate of reaction. And the way they do this is by decreasing the energy that's required to initiate the reaction. And that is called the activation energy, or the energy of activation, whatever you call it. It's the energy required to make a reaction go. So enzymes decrease that energy of activation, and in doing so speed up the rate of the reaction. Remember that enzymes are not going to be used up in the reaction. They can be used over and over again. Some enzymes require cofactors or coenzymes. Some do not. And then enzymes are going to have an active site that is highly specific for a certain substrate. The amount of energy required to initiate every metabolic reaction, so every chemical reaction that occurs has to have a little bit of energy put into it to initiate the reaction, and that's called activation energy. What is the sum total of all chemical reactions in the cell? That's going to be metabolism. And remember, metabolism is anabolism, or the building up reactions, the reactions that make larger molecules out of smaller molecules. So it's going to be anabolism plus catabolism. And catabolism is the breaking down reactions, where you take large molecules and break them into smaller parts. All right, review aerobic respiration. I want you to be comfortable with the concepts that we talk about in Chapter 8 when it comes to cellular respiration. So I want you to know, in prokaryotes, aerobic respiration is going to occur in the cell membrane. In eukaryotes, aerobic respiration occurs in the mitochondria, right? What's the point of aerobic respiration? Well, it's to make energy. And the mitochondria are the energy makers of the cell. Now, prokaryotes don't have membrane-bound organelles like mitochondria, so they have to use their cell membrane instead. Why do they have to use a membrane to begin with? Well, the membrane is where the enzymes of aerobic respiration are going to be located. So that's why we have to have access to some kind of membrane in order to perform aerobic respiration. In prokaryotes, it's going to be a cell membrane. In eukaryotes, it's the membrane of the inner membrane of the mitochondrion. What is the final electron acceptor during aerobic respiration? Well, go back and recall kind of big picture stuff. What does aerobic respiration do? We take glucose, and through a series of redox reactions, we take electrons from glucose and we shuttle it through the electron transport system, and in doing that, we generate ATP. Those electrons have to go somewhere. In aerobic respiration, the electrons go to oxygen. Okay. During which phase of cellular respiration is the majority of ATP produced? Well, remember, we have glycolysis, we have the Krebs cycle, and we have the electron transport system, or oxidative phosphorylation. And it's during oxidative phosphorylation that the majority of ATP is going to be produced. I would go back and really pay attention to Chapter 8 and Chapter 8 lecture. I try to go over this in as much detail as I can without getting too complicated. And of course, if you have any questions, just reach out to me and I'm happy to clarify. Okay, and then finally, an enzyme binds to its substrate via the active site. Remember that enzymes are going to have a highly specific active site that has a very unique shape that really only fits a substrate perfectly, okay? Okay, and then I've got some practice questions here for you. So, number 1, which of the following is a taxon that contains all the other taxa listed? So this question is basically asking you which taxon of these given taxa is most inclusive, which includes the most organisms. So we're going to look for the broadest taxon, which out of this list is going to be C, kingdom. All these other taxa fall under the taxon kingdom. Number 2, the term that refers to the presence of a tuft of flagella at one end of the cell surface is, and this is going to be going back to your flagellar arrangements, which you need to know. And the flagellar arrangement that involves a tuft of flagella at one end of the cell is going to be B, Lophotrichus. 3, which organelle contains cristae where enzymes and electron carriers for aerobic respiration are found? You need to go and review all of the organelles of eukaryotes and be familiar with what their functions are. The answer here is going to be mitochondria, and this makes sense because the point of aerobic respiration is what? To produce energy, or ATP. Mitochondria are commonly referred to as the powerhouses of the cell because that's where all the cell's energy is made, or the vast majority of the cell's energy is made. All right, the correct sequence of events in viral multiplication. Remember, I'm going to give you an example using the bacteriophage multiplication cycle. And so it's not going to have the uncoding step in it. So here, this is what the question is going to look like, and you need to pick the one that has the correct order. So that's going to be B. Adsorption always comes first. Then you're going to have penetration, replication, assembly. Those assembled virions or viruses are going to mature, and then they're going to be released. Number 5, in blank conditions, the cell membrane will shrink away from the cell wall, plasmolysis. So go back and review hypertonic, hypotonic, and isotonic conditions and know what the cell with a cell wall is going to do in each condition. This is describing a hypertonic condition for the cell to be in. Number 6, which term refers to all of the chemical reactions within the cell? Well, remember, this is going to be the sum total of chemical reactions, so it's going to be anabolism plus catabolism, and that equals D, metabolism. So that's exam 1. This study guide has topics over every single question that you're going to see. So watch this review several times, make notes on the study guide, review those notes every single day until the exam, and you should make a pretty good grade. And as always, just reach out to me if you have any questions, need any clarification over any of these topics. I'm always happy to help. I want you guys to make the best possible score that you can on this first exam. Really get off to a good start in microbiology. So anyway, good luck studying. And remember, you've got this.